Hi guys, welcome back to another tutorial video. In this video, we are going to discuss about the microbial production of glycerol or the industrial production of glycerol by microorganisms. As we came across, there is a lot of precursors molecule for the production of glycerol, which includes biodiesel, petroleum products, vegetable oils. But here we are going to discuss about the production of glycerol by microorganisms. Glycerol, as we know very well, also called as glycerin, it is a colorless, odorless liquid which has a sweet taste. Generally, glycerol is non-toxic and soluble in water and remains viscous at the room temperature. The molecular formula of the glycerol is C3H8O3. The glycerol that reacts with organic as well as inorganic acids, which results in the formation of many derivatives such as aldehydes, esters and ethers. As I said before, the glycerol also called as glycerin, glycyl alcohol, propone, 1,2,3-triol, etc. These are all the general introduction about the glycerol. History. Glycerol was first obtained from the process called saponification. So it's a byproduct which is derived during the soap production. So it was first discovered in the year 1779. Since last 20 years, it's been a primary source for especially for the production of fatty acids. Microorganisms for the production of glycerol. The common brewer yeast, baker yeast or the osmotolerant yeast, which is the sugar loving yeast, which are commonly used for the production of glycerol, which the examples which includes Saccharomyces cerevisiae, Candida versatilis, Candida magnolia, and some of the filamentous fungi, Aspergillus niger, Aspergillus oryzae, which are involved for the production of glycerol. The yeast that produces the glycerol by the pathway of glycolysis by reducing the intermediate molecule, which is dihydroxyacetone phosphate to glycerol 3-phosphate. These glycerol 3-phosphate undergo the process called dephosphorylation to yield the glycerol. So this is the pathway. By using the pathway, the microorganisms that produces the glycerol. The osmotolerant yeast that are commonly involved in the production of glycerol. So it uses the glycolysis pathway by reducing the intermediate dihydroxyacetone phosphate to glycerol 3-phosphate. This glycerol 3-phosphate, as I said before, it undergoes the process called dephosphorylation to yield the glycerol. The filamental fungi, as I said before, the Aspergillus species that uses the anabolic DHA pathway. Here also the intermediate is dihydroxyacetone phosphate. By using this intermediate, it produces the glycerol. The optimization of best fermentation conditions for the production of glycerol. Continuous oxygen supply and agitation are necessary. Raw materials such as sugar beet molasses. Carbon source includes glucose sucrose, which is in higher concentration because of osmotolerant yeast going to carry the process. Nitrogen source includes disodium sulfate, yeast extract, peptone, and dihydrogen potassium phosphate. Various source sulfate ions and NaCl. The best pH for the production of glycerol 6 to 7. The optimum temperature 22 to 32 degrees Celsius. The duration to carry the entire process takes 72 hours. The yield 82.3%. Purification and recovery process. Filamentous fungi that are involved in the production of glycerol. So the mycelia has to be extracted by the process called vacuum filter. Weak cationic exchange resins involved in the ion exchange absorption and treatment with the activated carbon for the removal of impurities. Applications: it is used as emulsifying agent, act as a antifreeze agent. As I said, the emulsifying agent widely in the food industries, used as also lubricant in pump and gaskets, and also used as solvent for flavors and food color, widely used in cosmetics in the form of moisturizer, heals the skin wound, reduce acnes, eye disorders, and vasodilator. These are the some common applications of glycerol.